Next speaker, all the way from Tasmania, is Barbara Holland, learning from diversity. There we go, that's a good start. Oh. Thank you.
Am I audible? Yes, I am. Okay, cool. Great, thank you. Yeah, so I just wanted to talk a little bit about some of the work, mostly going to be talking about work from people at UTAS, but there are sort of collaborators beyond that around the centre as well, from people that take a phylogenetic perspective on things and why that might be useful. Some of the work I'm going to be talking about or alluding to, you'll have a chance if you're here for the next 2 days to hear about in some more detail. So Ben, Luke and Jonathan all have talks on Tuesday, and Ali has a poster in the session that's tonight, which I strongly encourage you to check out. So yeah, so why should we care about phylogeny? Like if we're interested in, I guess, building better crops that are, more resistant or more capable of resisting sort of arid conditions. What is it that a sort of evolutionary perspective might help us with? And so I've sort of picked out 4 things that I guess are sort of examples where I think phylogeny can help. So one is just sort of understanding origins of traits. Another is sort of, given some set of traits, how have they co-varied over, long sort of swathes of evolutionary time. Also, I guess this sort of idea of phylog to P or phylog-GWAS, as it's sometimes called, where you're trying to basically associate some region of a genome with a phenotypic trait of interest. I'm embarrassed after the previous talk to say that none of the methods I'll talk about will really cope well with copy number variation, but maybe some of what Jonathan will talk about might touch on presence-absence data. That's a hint, Jonathan. Make sure you have a slide in your talk that's something to do with that. Right, yeah, so origins. So I mean, I guess one reason you might care about trees is just blatant curiosity. So Origin of the Species got published in, I think, 1859, according to my slide here, and just a little bit more than 5 years later, Ernst Haeckel, for his PhD, published A Tree of All Life. So if you're worried, your PhD project isn't ambitious enough, maybe it isn't. Maybe you should try and be more like Ernst Haeckel. But I had my sort of couple of sort of influential mentors when I was back at Massey University, Dave Penny and Dave Lambert, and they'd worked a lot on phylogenies, but both of them really passionately said, well, we don't really care what the phylogeny is. I remember Dave Lambert once said, if someone published A Tree of All Life and put it on a poster outside my office, I wouldn't even bother to look. It's like, well, maybe I'd check if mammals were monophyletic, but it's sort of like, the reason that you actually want to know the tree isn't so much because you care about the tree, is because it can tell you something interesting about how some, trait has evolved. So, and Dave Penny always used to say, I just want to get the tree solved so I can get on and answer the more interesting questions. And so I just chucked up a couple of random examples, not particularly planty at this point, but so the one on the left there is sort of simian immunodeficiency virus, and so this was an interesting one to me. I always assumed HIV was just something that had happened once, but it's actually, you've had zoonoses on at least 4 separate occasions according to that phylogeny, which is sort of interesting. The one on the right is a tree of sort of mitochondrial genome variation that sort of really, really strongly supports the sort of out of Africa hypothesis, or one of the sort of main supporting sort of pieces of evidence for that. You've got all of these sort of genomes here below this line here are African ones, and this is the diversity on the whole rest of the planet, basically. So it sort of kind of sort of tells, this tree tells an amazing story about human origins. And I had to put one slide in as a nod to my PhD supervisor, Dave Penny, who sort of sadly passed away just about sort of 3 weeks ago or so. One of the pet reasons he loved trying to work out the phylogenetic trees of mammals and birds in particular is he got really, really annoyed whenever anyone said, this classic story, asteroid hits the earth, dinosaurs go extinct, mammals and birds, you know, expand and inherit the earth. And he's like, well, what evidence is there for that really? That's just two things that happened at the same time. How can we know one really caused the other? Even a first-year stats student knows that, you know, correlation doesn't imply sort of causation. So he was sort of saying, well, you know, if you actually have dated molecular phylogeny, you can really test this hypothesis. If it really looks like this, then yeah, maybe the asteroid story is true. But if you're finding that you've got mammals and bird origins coming back way before the KT boundary, then not so much. But I love this quote of his that, you know, he sort of hated this. It's a Marxist view of evolution because it denies a role for competition. So I thought, yeah, it was fantastic. Yeah, so in the plant setting, so we have here, so this is a tree of about 600 different eucalyptus species. And one of the projects that we've been sort of, I guess, interested in, both at, I guess, the sort of people at Hawkesbury and at UTAS, is sort of adaptation to arid conditions in the eucalypts. And so one of the things that you can sort of see if we're plotting this as log moisture, I think, on here, and each of these little red dashes here is essentially an phylogenetically independent transition from a wet into a dry environment. That's kind of interesting because if that had only happened once, then it would be, in a sense, like really difficult. Even if you had a lot of species that were dry adapted, if it's only happened once, you're not going to have much power to tell, you know, what was associated with that change, because in some sense you've only got a sample size of one. But here, the sort of the phylogeny is telling us, no, this is a transition that evolution has sort of successfully accomplished on, you know, maybe 20 different occasions. And so once we know that, we can start to look for, well, what are the traits or suites of traits that have sort of shifted in some coordinated way with that change to an arid environment. And so that's work that Ben has been looking at amongst sort of other people. Yeah, and so phylogeny is obviously like in some sense, like the two Davids were saying, it's kind of the ultimate nuisance parameter. We need to know it because it's sort of a confounding variable. So if you imagine you had, I'll point at this screen now to be kind to the people on this side of the room. So you have two traits, sort of X and Y. You just do sort of a simple scatter plot, you can see they're correlated, and you might think, oh, there's something really interesting going on here. But if it so happens, you know, that this was your phylogeny, and this is all one clade, and this is all another one, then you can basically see that correlation is essentially sort of illusory. So if you don't really know something about the phylogeny, if you aren't able to sort of somehow to account for it in your statistical analysis, then you could sort of come to some wrong conclusions about what traits really sort of go together. And so Ben and Luke have sort of been doing a lot of work on just trying to work out what are the most appropriate techniques for kind of dealing with this. It's a very long and complicated literature around comparative sort of phylogenetics. And I think they've at least convinced me, and I think hopefully some other people as well, that these sort of multiple response phylogenetic mixed models are a beautiful way for looking at how traits co-vary. And so, whoops. Wasn't what I meant to click. I meant to click this. Yeah, so these models, you can basically sort of encode the similarities of taxa in a tree that's going to affect how correlated you expect traits to be. If things are sort of phylogenetically similar, you just expect their traits to be similar because they haven't had long to diverge. But also, you know, some particular traits might just naturally stay coordinated more over long periods of evolutionary time, whereas others you might be sort of tinkering with sort of at the tips of the tree. And so these methods basically allow you to decompose the correlations you see amongst traits into those that are being conserved over sort of phylogenetic sort of evolutionary timescales and those which are more sort of pliable, I suppose. And they've got a nice paper out with Mark Westerby. And there's this one, which I think is almost going to jump across the review process anytime soon, but is already up on bioRxiv and has a fantastic GitHub software tutorial on sort of showing how you can kind of implement some of these methods in some nice R packages. And so, yeah, so Ben sort of applied some of these techniques to eucalyptus and been able to see, well, what are the traits that have just sort of stayed associated in the same way over the whole tree, what are the ones that are sort of more sort of malleable. So we've got this sort of phylogenetic correlations, and he's looked at, so things like, wood density, height, leaf area, but then also just environmental things like amount of water, temperature, and some soil traits like bulk density and perhaps something to do with potassium. And you can see some of these things have stayed coordinated over long amounts of time, whereas others you see sort of residual correlations. And I mean, that's potentially interesting in the context of, sort of Charlie's talk from earlier today, where if you're sort of thinking, evolution's had a lot of time to try and find optimums, and if there's some things that it hasn't been able to shift, that have just always stayed coordinated for the last 80 million years, maybe they aren't the things you should be trying to, make combinations of traits of. Maybe you should rather be sort of, I guess, tinkering with the stuff that you can get these different residual correlations. And then, so another sort of area that we've been looking at is this sort of philo G to P, so loosely trying to associate genomic reasons with particular traits of interest. And so here, say we had a trait that's happened multiple times, and it could be anything really. Here I've just used, say, wild, relative, or domesticated crop as an example here of something that could be an independent transition. But one of the things you might be interested in is there any variation in the edge lengths on your phylogeny that's sort of associated with that trait. And you certainly don't expect the whole genome to be sort of doing something that's correlated with a trait. Like probably most of it's just doing, some kind of background thing where the patterns you see in some sequence alignment kind of just mostly match the patterns of relationships you have in your tree. But what you'd like to be able to do is sort of pull out particular regions, or so genes or parts of genes, where maybe you see rate variation that correlates with that trait that you care about. And so I would say that sort of the common existing phylogenetic methods aren't super well equipped to do that. So I guess phylogeneticists have sort of understood for ages that there's lots of rate variation that you need to account for when you're trying to build phylogenetic trees. But the two ways they've mostly done it are sort of so-called partition models, where you basically sort of decide in advance, so this is a gene boundary, so I expect this to maybe do something different from something else. Or mixture models, where you basically say, it could be, I don't really know exactly what my edge lengths are. Maybe there are some sites that are evolving slowly and some that are fast and some that are doing sort of different mixtures of things. So I'll just fit a profile of trees with different edges to my whole sequence alignment. But that often ends up being sort of very over-parameterized and hard to interpret. And so one of the things that Ali has been having a look at is this idea of phylogenetic hidden Markov models, which I think are sort of an attempt at getting the best of both worlds, where you don't have to say in advance, I think this particular region, I'm expecting it to be doing something different, like you do in a partition model. But you basically use a hidden Markov model to say, I've got some hidden state that I've got some regions that are maybe like this and other regions that are maybe in a background state, and I'll just sort of try and pick out using sort of Viterbi algorithm, HMM ideas, like which regions are which. And so we're getting some sort of preliminary, somewhat promising results with those ideas. Last slide, which is good because I can tell I'm running out of time. Phoebe isn't in tackle mode quite yet. No, okay, good. So basically the sort of a phylogenetic approach to designing this comparative transcriptomics experiment we've been sort of running over the last few months at UTAS. So Ben was sort of responsible primarily for the sort of species selection, where we've got 8 different pairs of species where one's more wet adapted and one's more dry adapted, so looking for basically contrasts, but they're otherwise sort of fairly close, maybe not sister species, but closely related species, and sort of growing them all up in sort of common trays and common conditions, with one sort of set getting multiple drought treatments and another set being well watered. So Chris will tell you more about it, but then I'm hoping Luke will also tell you a little bit more about how we're planning to try and analyse that data mathematically. So obviously, you know, the typical sort of gene expression study, you might want to know are some genes, you know, doing something different in drought conditions to non-drought conditions. But we're also hoping that, so that's already interesting, but if you've the same gene is doing something different across 8 different species pairs, then you're like a lot more interested again. And if that same gene is doing something more different in your species that are, you know, known to be drought adapted, then that's again, you know, maybe more interesting. So we're looking for effects and interaction effects. And so, yeah, so hang tight for future talks by Ben and Chris and Luke on that topic. I have 9 seconds left. I'm shutting up right now. Oh my God. This countdown time is alarming.
Thank you, Barbara. We're going to allow questions, are we, Phoebe? Oh, you can tell who's in charge.
So another interesting area to use phylogenies, which I think is really relevant to this group, is gene transfer. So domesticated species very often transfer back and forth with their wild progenitors. And you can imagine among some eucalypts, you might have multi-way species.
Adaptive sort of
transfer.
hybridization and things like that. Yeah.
So there's the Abba-Baba test for a trichotomy is well worked out. But as far as I know, no one's worked out extending Abba-Baba to much larger phylogenies. And that may be something interesting to think about.
I didn't actually have that slide in there, but it is a little bit like what Jonathan is trying to do with his convergence-divergence models, is trying to sort of expand that idea. So I would say, Jonathan, wave. Yeah, come and have a chat with Bruce at some point. We can.
Okay.
Yeah.
Excellent. I'm glad we asked and answered that question. I think we better move on. Thank you so much, Barbara. Very entertaining.